Alldus podcast is brought to you by Alldus International, supplying your expert AI and digital transformation staffing needs across the US and Europe. Today, you are listening to our AI in Action series, where leading minds in AI from across the world share their story, success, and advice. AI in Action cuts through the hype and explores the true impact of artificial intelligence in our world today.
You're listening to AI in Action. I'm your host, JP Valentine. Our guest today is Brandon Allgood. Brandon is the Chief Data Officer at Fog Pharma. Brandon, welcome to the show.
Thank you for having me.
So Brandon, let's start with yourself, please, as we do with all of our guests. Could you please give us a bit of an overview of your background in technology, from where you got started, some of the roles you've held along the way, and what's led you to where you are today as the Chief Data Officer for Fog Pharma?
My PhD is actually in cosmology. Cosmology was one of those topics that really excited me, the involvement of math and large-scale computing to really understand the fundamental state of the universe, understand the fundamental laws of the universe. So I spent a lot of graduate school writing software, writing machine learning algorithms to analyze things like images from Hubble Space Telescope and other types of deep galactic imaging. And throughout graduate school, again, the plan was to be an academic, but towards the end of grad school, decided that academia wasn't quite for me, at least not right away. And having been next to Silicon Valley, it was pretty obvious my next move was to jump over the hill and apply my AI, mathematics, and software engineering skills that I had at least begun to learn in graduate school. And I say begun to learn because I learned very quickly in my first job that I was not nearly as prepared for the things that I would end up doing as I thought. Out of graduate school, I received an offer from Google, NASA Ames in Silicon Valley, and a small startup. NASA Ames felt a little bit like academia, and so was not as excited. That was a safe route out of graduate school. Google at the time was not the Google we know today. This was back in the early 2000s, and ad serving and developing machine learning algorithms to serve ads was less than exciting to me. Looking back on that, I would have been a lot wealthier today had I gone that direction, but c'est la vie. I tend to be more scientific and less wealth driven in my life. So I ended up joining a small startup that was doing super interesting things in the area of chemistry, machine learning, and at the time, large-scale computing, which meant Beowulf clusters. For those of you that are quite old, for those that don't know what Beowulf clusters are, look them up. Beowulf clusters and hypervisors are ultimately what led to the cloud. But yeah, they were doing interesting things in chemistry. I said, I'm a physicist. Chemistry is just physics. So I jumped into that, learned a lot about what not to do in a startup company. We did a lot of things quite well in that we developed a lot of really cool early technologies of applying machine learning to the problem of drug design. And this is in 2005. But in terms of building a company, in terms of CEO, board, other things, I think we made a number of different mistakes. Ultimately, that company ran out of runway, ran out of money. And we turned around and I then co-founded another company called Numerate, where really same technology, same kind of approach to solving the problem of drug discovery using machine learning. And at this point, it was 2007, AWS was around and we immediately launched on the cloud. And I believe for a while there, we were actually the biggest users of AWS. This was really early on. We were breaking their APIs left and right. Netflix ultimately eclipsed us. And yeah, from there, built up Numerate over many years, tackling many different problems within drug discovery and development, and built up a software platform. Then ultimately started another company called Valo Health, merged the two companies. Effectively, Valo acquired Numerate. And so at Numerate, I was the chief technology officer. At Valo, I ended up being the chief AI officer. Again, different titles. It's just more a reflection of people's recognition of what machine learning can do and its impact. Back when it was Numerate, it was just chief technology. Then it became Chief AI Officer, which was more of a buzzy word. I've never been a title person. At Valo, we broadened the capabilities from small molecule design to large molecule design, and also began to apply machine learning and things like target identification, more biology-centric things. We started to bring in real-world data, human data, both in terms of optimizing clinical trials using machine learning algorithms, but also using human data to better inform disease models, which ultimately became drugs. Then took about a year off. This was a year and a half ago, about a year and a half ago, I left Valo as an entrepreneur. I had worked 60-hour weeks since I left graduate school, and I will openly admit that I'd burned myself out, and so I needed to take a year off. I was a ski bum for not this past winter, but the winter before that. That was refreshing. And then I started getting itchy again, and I started a consulting firm, investment firm, where I was working with large companies, working with venture capital, working with small companies, investing some of my own money in some of the really early stage companies, just working. And one of the things that I got excited about were peptides as a therapeutic modality. And then ultimately looked around at different companies and that were working in peptides, and realized that Fog Pharma was definitely one of the leaders in peptide design. Luckily, Amitai Mammon, who was head of R&D at J&J, he and I have known each other for a very long time. He asked me to come and consult with Fog Pharma. Ultimately, I fell in love with the team, with the technology, and ended up joining the team as the chief data officer. And so that's maybe a bit more detailed than you normally get, but that's my journey from cosmology through to where I am today.
Brandon, thank you for sharing that, and thank you for going into the level of detail, because your background and journey is quite interesting. You've touched on it there a little bit. Let's talk about Fog Pharma. You've set the stage, leader in peptides, but for the audience listening who are not familiar, tell us all about Fog Pharma, who you are, what you do, mission of the business, and help really set the stage for what it is you are doing as the Chief Data Officer.
My passions are large-scale computing, machine learning, and science. And I've spent most of my life in small molecules, but also in, like I said, biology and real-world data. When I took time off, I started to see these interesting things happening in peptides. And peptides as a therapeutic modality, we've had peptides as drugs going back 40 plus years, but for the vast majority of that time, the peptides that we have as drugs were either directly natural products, i.e. they came from a sea sponge or some other sort of derivative of a plant, or it was some modification of a natural product. Peptides on their own are quite complex, so they are larger than small molecules, more complex than small molecules, obviously smaller than proteins, but in an interesting way, they're more complex even though they're smaller than proteins. And so what was really interesting is, I think, when looking at what was happening, and it's this confluence of technologies that have been developed for antibody and protein design. So the synthetic biology, the ability to build and rapidly build peptides and actually be able to build gigantic libraries of peptides to be able to do large scale screening. That technology has been being developed over the last 10 years. That paired with machine learning, and machine learning being able to start to begin to do generative work in order to think about peptide design, think about the various properties of peptides. The combination of those two things, I would say, are the killer combination. So much so that I think peptides have the ability to do things that small molecules and proteins can't. And there's so many targets or so many proteins that we know are dysfunctioning and leading to disease, but we can't go after them with small molecules or proteins. But I do believe a big class of those will be opened up by peptides, which means that peptides as a therapeutic modality could be as big or bigger than small molecules. That's my belief. I'm doubling down on that here. And when I was started to look at the different companies out there, I realized, and the technologies that these early peptide companies and some larger pharmas are also working in the space, I realized that Fog had a really interesting idea and a really interesting modality, in that the observation of the founding CEO of the company, Greg Verdine. So one of the big challenges with peptides is peptides are often not unable to get into the cell, at least engineered peptides, and or bind. And Greg had an observation that we know of peptides that go through the cell wall. We know of peptides that bind, and those peptides are generally what are called alpha helices, so tight corkscrew sections of protein. Those are the types of proteins that go through the cell wall, happily sit in the cell wall, and proteins, extracellular, intracellular proteins connected. It's this alpha helix. And also, when you look at protein interactions, an alpha helix, this corkscrew, is often involved. And the problem is that when you take 10 amino acids or 20 amino acids, a protein is generally longer, like 100 plus amino acids. When you split out just the alpha helix part, it unravels. It doesn't stay in an alpha helix. And so interestingly, what Fog, or Greg and in his lab at Harvard and at Fog, developed is a stapling technology where they literally have these molecular staples that keep these peptides in an alpha helical shape, at least from a kind of a statistical mechanics perspective. So these alpha helices are now what we call helicons, these stapled alpha helical peptides. Those actually go through the cell wall relatively easy. It's not always the case. It's not perfect. We actually use machine learning models to predict and to generate helicons that have better improved cell penetration. But then also those alpha helices naturally bind to proteins. And so that was really interesting to me. On top of that, I found that the group, in terms of the teams and the executive team, that they were, having observed them as a consultant, I was super excited that everybody was collegial here. It was a wonderful culture at Fog. And then on top of that, the third thing is, when I often joke, there's a lot of shiny things in Cambridge, in and around Cambridge. And there are a lot of companies that are formed around those shiny things, but most of those companies are going to fail. And the reason is that they're going to fail is not because the shiny thing didn't work. The shiny thing was probably just fine. They will fail because they do not know how to execute. And one of the things that I really liked about Fog is not only did they have this platform that they were building, but they had already advanced a molecule from zero to the clinic. And it was, and so Fog 001, which is our first clinical compound, was already in humans. And at the end of the day, whether the platform works or not, I could tell you, the team knew how to execute. They knew how to go from zero to the clinic. And I think that's often a rare thing to see. Kind of those combinations, I got super excited about Fog, the technology, the people, the company, and I went all in.
You are listening to the Alldus podcast. When you're looking to scale your team, or if you are interested in showcasing your company in a future episode, reach out today. Or if you're in the market for a new role, visit our website to view open positions, www.alldus.com.
I want to jump straight into your role because you're leading the data science and engineering side of things. For people who are in the biotech space, a lot of what you're talking about will resonate. For those listening who are maybe not as familiar, can you talk about how data science, machine learning, and AI can accelerate this research, what it can do to bring novel treatments to market, and how combining it with the space of peptides, it's unique in nature? Walk us through your role and responsibilities and how that all pulls together.
I think there are tons of aspects, right, where machine learning, data science can help. So let's start really at fundamentals, right? So designing a small molecule or a peptide to ultimately become a drug, that is a high dimensional optimization problem. So what you're saying is that we want to design a molecule that can be active against the target of interest, do the thing we need it to do at the protein level, has to get into the cell. You have to be able to get it into the blood. It has to be metabolized not too quickly, not too slowly, Goldilocks zone. It can't bind to anything else, meaning which potentially may cause toxicities. So there's a laundry list of properties that you want to get out of, that you need to have in this molecule in order to make it a drug, in order to be able to be confident that you can put it into a human, that the human that it won't cause undue harm to the human, and that will actually make its way to the place we need it to make it to, and it will do the thing that we need it to do. And so high dimensional optimization problems are key places where AI can be brought to bear in order to find solutions. So not only is it a high dimensional optimization problem, so the interesting thing is, this is kind of funny. We used to talk a lot about the space of all potential small molecules that could be made. And the space of all potential small molecules that could be made is 10 to the 60. So 10 to the 60 60, and that's gigantic, right? You cannot enumerate that. You cannot even begin to think about search algorithms that can really effectively search that, not only search that with multiple criteria. And so this is a huge daunting problem. It turns out with helicons or these alpha helical peptides, the space is even bigger than that. And so think of an infinite space. And what you're trying to do is find the needle in the haystack in that combinatorial space. It's not a smooth space, it's a combinatorial space. And so this alone, just that, you can think this is a prime place where we're going to have to bring machine learning to search that, because humans generally, when they do this, they optimize one property at a time. So maybe they design the molecule and they optimize affinity or activation against the protein. Then they start to optimize, can it be soluble in liquid? And as soon as they change, every time they change an atom, they affect the other property. So it's not like these properties are independent. And so building machine learning model, the human brain at any given time can maybe think of a few properties or along a few dimensions at a time. A machine learning model theoretically can think along hundreds of different axes at a time. The real problem then becomes, do you have the training data in order to train the models, either supervised models or generative models, such that the model can understand how to make changes along those different axes? And so there, the answer often is in this space, no, we don't have enough data, right? This is a very different space than large language models, where the data is literally being produced by us humans on the internet constantly and for free. Here in our space, a single data point can cost you $100, $1,000, $10,000. And just by that measure alone, and could take 3 months to generate, by that measure alone, we're never going to have the right amount of data. So then we also have to think about not only supervised and generative models and how to build them, but how do we also develop active learning loops? So actually almost go to the meta level and think about, how do we extrapolate out of sample, then go and make and test, and then extrapolate again out of sample, and do that in a way where our extrapolations are as far as we can get away from the training set, because generally you're shooting, if you had what you needed in your training set, you wouldn't be doing this in the first place. But project out and do that with high confidence, then go and make and test, and really tighten that design, make, test cycle with machine learning. And so there's a lot of things that we can think about. And the interesting thing about peptides more directly is that with small, the world's been looking at and pursuing small molecules for 50, 60, 70 years, at least quantitatively. And we have data and people know how to do this. Proteins are a little newer, but I think there's still a lot of data out there within databases. Peptides, there's nothing. And it's almost this green field both in terms of data generation, but also in terms of our understanding of how to model it. What's the right featurization for small molecules and proteins? I think we're starting to figure it out. With peptides, it's a whole new field. How do we think about feature generation and what's important in the properties to these different aspects, to these different dimensions that we're trying to optimize for? So tons of stuff there. And that's just in Helicon or peptide design. Once you go outside of that, there's also a lot of questions around the biology, around cancer biology, which is where Fog primarily focuses. We're focused primarily on deadly cancers. And so we're focusing on those cancers and those diseases where patients are suffering and ultimately dying. And we're trying to design drugs to prevent that. And how do we also then look at the biology of disease, look at the biology of cancer, and better understand and predict connections between proteins? So there's a lot of graph-based machine learning there. And then now in the clinic, we're looking at patients and trying to understand patients and their interaction with our drug? Can we predict which patients, based on their longitudinal patient journey and their genetics, can we predict which patients are more likely to respond to our therapy, and which patients aren't? And if they aren't, are there other things that we combine with our therapy, like PD-1 inhibitors or others, to then go after that? So there's all sorts of different things we can apply various levels of supervised, unsupervised, and generative machine learning to.
Brandon, thank you so much for the level of detail. Normally, I would ask what the exciting roadmap looks like and the potential use cases, but you covered it all there. So I want to finish on my final question, which is always the platform for you to speak to an audience of potential future employees. As someone who made a conscious decision to switch from the more traditional biopharma space into peptides because of your interest and your belief in what its capabilities can be, speaking now to an audience who may not have made that switch, may not have thought about it before, what could you tell them about Fog Pharma, the near-term growth roadmap, and why they should consider working with you over some of the other more traditional biopharmas in the space?
Yeah, so it's a good question. This could actually even pertain to any of the companies that I've started. I won't work at a company that's not taking a data-driven approach to drug discovery and development. One of the things you could say about Fog, but any of the other companies that I've helped build or worked at, is that we take data as the primary currency in which we deal, and how we ultimately get to drugs that ultimately benefit patients. It's a little different here in Fog, in that Fog's been around for about 8 years, and data science is relatively new. Data science was brought in about 3 years ago, but with the transition from Greg to Matai, we've really doubled down on this idea of data and of how machine learning and AI can propel all aspects of the business. So we are thinking about solutions that go from using large language models for internally generating documents, thinking about using large language models for regulatory documents and those types of things, to using novel machine learning algorithms for target ID, again, through the Helicon design systems that I talked about, and into the clinic. And we're already also thinking about, if Fog 001 ultimately becomes a commercial product, how are we going to use AI to ensure that those patients that can benefit from our drug get our drug? And so all I would say is that these are, for all that vision, we're still early on in the journey. We've developed a bunch of really cool technologies around Helicon design. We're starting to develop similar things in computational biology and real world data. But ultimately, we want to build a computational platform. And by platform here, I think, at least within biopharma, platform has this kind of amorphous definition. And when I say platform, the combination of the lab, the complete integration of the lab and the machine learning, really, and building a system that covers in one kind of system the end-to-end target ID all the way through commercial. And that's an audacious thing to build. It's going to take a lot of engineering. It's going to take a lot of data science, and it's going to take a lot of time, because unlike building traditional software, there are a lot more unknowns. There are unknowns about peptides and how they operate. There are a lot of unknowns about how things are going to operate in the clinic. So we take a slightly different approach, but for me, a disciplined engineering approach to how we push things forward. And at the end of the day, I also think about data science as ultimately becoming an engineering discipline, meaning I think in a lot of companies, the norm is for the bench scientist to push the data scientist button. To ask a data scientist, can you do this analysis? Can you do this? Can you do this? And ultimately, that's inherently human intensive, and which means it doesn't scale. And so as we start to build the platform, we want to be able to really not only design these systems, but actually bring them all the way to the bench, such that The experiment is happening next to the system, or completely integrated with the system, that is also building and deploying these models and ultimately these designs. So the data scientists in many ways can move on to the next big project, and the system itself can scale, and ultimately more and more patients benefit from our drugs.
Brandon, thank you so much for coming on today and talking to us. Really appreciate you sharing your backstory, fascinating journey with many interesting steps along the way, but most importantly, the insight into the work that you're doing now at Fog Pharma. Peptides obviously getting a lot of publicity and interest within this field, and it's great to hear how AI and data can help accelerate the research so that we can all benefit from some of the possibilities that it can hold for us. So we're grateful for your time. We wish you, the team, and everyone at Fog Pharma the very best of luck in the coming months. We look forward to having you back on the show again in the near future.
Yeah, thanks for having me, JP.
Thanks for listening to this episode of the Alldus podcast. If you enjoyed today's episode, don't forget to subscribe, rate, and review. We are available on Apple Podcasts, Spotify, and any Android podcast of choice. You can also head over to our website, www.alldus.com, to listen to more podcasts, view our open roles, and stay up to date with industry news. Thanks for listening, and stay tuned for more great episodes coming very soon.